Hello, good morning students. In the previous video, we discussed about the structure of microsporidia. Now let's discuss the process of microsporogenesis. In the structure of microsporidia, we discussed that microsporidia is made up of four layers. Outermost epidermis, endothelium, middle layer, and innermost layer is tapetum. Now, in the process of microsporogenesis, we will know about how the microspores are formed. The process of formation of microspore is known as microsporogenesis. When formed during the process of microsporogenesis, the innermost part, that is the sporogenous tissues, which is covered inside the tapetum. This is the tapetum. Inside the tapetum, all the space is filled by the sporogenous tissues. These sporogenous tissues give rise to the pollen mother cells. As in the previous video, I told you these sporogenous tissues, all the cells of sporogenous tissues are able to form microspores, but some cells get degenerate and some take part in the formation of microspore. Sporogenous tissues earlier, when the anther is young, these are tightly packed. The cells are present very near to each other, but as the anther grows, its size increases, these cells of the sporogenous tissues are arranged in a loose manner. Means they are loosely packed now when anther becomes mature. For the formation of microspores, the sporogenous tissues give rise to pollen mother cells. Means through the pollen and sporogenous tissues, pollen mother cells arises. Pollen mother cell arises through the pollen mother cell arises through the sporogenous tissues. This pollen mother cells, which is diploid in nature, it's diploid in nature. This cells goes through the division, and the division is meiosis. It takes place, meiosis division takes place here. And due to the meiosis, it forms four microspores. Four microspores are formed here, which are arranged in the form of tetrads. Four microspores are arranged here in the form of tetrad. The arrangement of the tetrad can be different in different families of the plants. Like in some cases, the tetrad is formed in such a manner. This type of tetrad is known as tetrahedral tetrad. Or it could be like this. It could be like linear or T-shaped. This is tetrahedral tetrad. This is isobilateral. This is linear arrangement. Linear, and this one is T-shaped. Out of this, tetrahedral tetrad is the most commonly used type of tetrad, which is found in angiosperms plants. In tetrahedral tetrad, four microspores are arranged in such a manner that we are able to see three from that side. One is found just behind these cells. So four microspores are arranged, and all these microspores are and in nature. They are haploid structure. Microspores. These are microspores which are haploid in nature. These microspores are arranged in the tetrahedral manner. We will discuss about it. Tetrahedral tetrad, when anthers mature, these microspores get separated from this tetrad and form four microspores. These are the microspores, which are the first cell of the reproductive structure, or it is known as male gametophyte. Microspores are known as male gametophyte. Male gametophyte. It is known as male gametophyte because it contains the male gamete, which is which take parts during the process of fertilization. Out of these microspores, they take part in the reproduction process. These microspores are now ready to develop into the pollen grains, and we will discuss the structure of pollen grains. For the formation of pollen grains through these microspores, for the formation of pollen grains from these microspores, this process is the formation of pollen grains. For the formation of pollen grains, structure of pollen grains. When these tetrads get separated from each other, all these four microspores get separated from the tetrad, these are now able to convert into the pollen grains. So we will discuss structure of each pollen grains. For the formation of pollen grains, for the structure of pollen grains, the enzyme is formed outside the pollen grains by the tapetum. Tapetum, as you know, we discussed about in the layers of the microsporangia. Tapetum, which is the innermost layer, it's nutritive in nature. Besides giving the nutrition, it also helps in the formation of enzyme. This is the microspore, and this layer is the plasma membrane of the microspores. Outside this plasma membrane, it secretes one more layer, that is intine. Outside the intine, one more layer is formed. Outside the intine, one more layer is formed, and this layer is not formed in the continuous manner. Some spaces will remain there. This is the enzyme, which is formed by the tapetum. This is intine. This layer is intine, and this layer is enzyme. So we can say that in the pollen grains, two layers are present. Outer layer is enzyme, inner layer is intine. Intine, that layer is formed from the intine. That layer is formed from the pectin and cellulose. Formed from the pectin and cellulose. And the enzyme is formed from a very special chemical substance, that is sporopollen. Sporopollen. The sporopollen is known as hardest substance. It do not get regenerate due to the effects of acids, alkalines, high temperature, or due to the biological decomposition. That's why it is known as highly resistant, or it is known as hardest substance. For the fossils formation of the pollen grains, the sporopollen play a very important role. We find the fossils of pollen grains due to the presence of this sporopollen. This sporopollen is help in the formation of enzyme. Enzyme is formed from the pectin and cellulose. Inside this, the nucleus is present, that is the single nucleus. After that, this nucleus also divide by the mitosis. This nucleus do mitosis. Nucleus divide due to the mitosis, and due to the division in mitosis, due to the division of mitosis, the structure that is formed is a binucleate structure. Means two nucleus are present in one pollen grains. This is intine, then we draw the enzyme, and the gaps will again remain there, and the nucleus which do the mitosis, and after mitosis the structure is formed is the binucleate structure. Two nucleus are formed here. After the division of the nucleus, the cell wall formation takes place. The cells take some cytoplasm and they form two cells, but their cell wall formation is not in the regular manner. One cell is formed is some little bit greater in the size or larger in the size. That cell is known as vegetative cell. The cell which is larger in size is known as vegetative cells. Another name of that cell that is used is tube cell. It can be also known as tube cell because it helps in the formation of germ tube during the process of fertilization. Second cell, which is smaller in size, we make it like this, but after that it will become like a spindle shaped structure which float inside the cytoplasm of vegetative cells. This cell is germ cell. Germ cell. This cell is germ cells which give rise to the male gametes of the flower. Vegetative cell, which is larger in size, it contains large amount of the cytoplasm, and the cytoplasm contains very much nutrients so that it can give nutrition to the developing male gametes. But germ cells, which is spindle shaped smaller size cells, its cytoplasm is transparent type. After that, you can see that we make some gaps in the exams. What are these gaps? These gaps are known as germ pores. These gaps are known as germ pores. During the exime formation, the deposition of sporopollen is not continuous. Sporopollen, as we discussed, is the hardest substance, so it's not easy to break this sporopollen. That's why the sporopollen is present at some places, and these places are known as germ pore. Germ pore are the place by which the tube cells makes tube, and this tube generate or emerges out from the pollen grains through these germ pores. After that means when fertilization process takes place, the tube is formed and the tube emerges out carrying the male gametes by these germ pores. When we make a furrow-like structure, a pore-like structure, then it can be known as germ pore. It's germ pore, or it could be like a furrow-like structure. Then we call it germ furrow. So this is the structure of microspore or pollen grain, which have two cells, larger size cell, that is the vegetative cells, smaller size cell is the germ cells, two cell trace. It's this type of structure or this type of stage is known as two cell stage. This is the two cell stage of the microspores. So in the almost 60% of angiosperms family, these pollen grains or these microspores are shedded into these two cell stages. The rest all the families of angiosperms, these generative cells get divided and it forms two male gametes. Overall, we can say that cells will become the three cell stage. After that, we will discuss about that how these pollen grains are helps in the later on. This is the structure of pollen grains, and these pollen grains help in the pollination and then fertilization process. Pollen grains are rich in nutrition. That's why nowadays the pollen tablets are used to provide the nutrition by a commercial manner. Some athletes, sports persons which require large amount of nutrition, the nutrition can be given by taking the pollen tablets. So it can be used to take the nutrition because they contain large amount of nutrition. Some plants have the pollen grains which can create allergies. Pollen grains have this very small size structure or small powder-like structure which can create allergies in some persons. That type of allergic conditions can be created. One example of such allergy is hay fever. Hay fever is a disease which is created due to the allergy of the pollen grains. The special example of these such kind of allergic pollen grain is the Parthenium. 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 It is also known as carrot grass. Has the pollen grain which create allergy in loads of the people, and that's why the pollen grains are allergic for the nature. This is the structure of pollen grains. After that, we will discuss about the female reproductive organ, and then we will discuss about the fertilization. Thank you. Keep studying. Keep watching.